Dr. Pankaj Bharmoria. Yeah.
Hi everyone. Can you wait?
Okay, and let me introduce you. Dr. Bharmoria has affiliation of Chalmers University of Technology, Gothenburg, Sweden, and he'd like to present his talk named Protein-Based Biomaterials from Biocatalysis to Photonics. Yeah. So
Well,
please.
thank you, Doctor, for the nice introduction. So my name is Pankaj Bharmoria. I'm a Marie Curie Fellow at Chalmers University of Technology. And today I'll share some of my research with you, that is protein-based biomaterials from biocatalysis to photonics. Well, this is the abstract of my paper that I personally think that proteins that do play as God in the biological system, as they perform a lot of duties from the generation, operation to the destruction of the life by changing their secondary structural conformations. So in this talk, particularly, I shall be highlighting the fact that how by changing the secondary structural conformation of the proteins in vitro can result in a new material for the desired application. Well, this is an example of how the proteins that play as God in the real life. Actually, proteins were actually the first living material generated on the earth. So in that term, they can be called as the generator of the life. Then operator of the life, proteins in the form of enzyme, they play a lot of duties in the daily life of biological systems. So one of the example is the transport of molecule across the cell membrane via the transport proteins. And another one is the destructor of the life. In the daily basis, we see that the bacteria decomposes a lot of food and many things, and they decompose it because of the enzymes, which are also proteins. So in that terms, they are called as the destructor of the life. So this is the conformation of a protein. Basically, the proteins are synthesized in the ribosome as a single polypeptide chain, and after that, it undergoes further processing and folding into a molecule, actually, which is this one. However, this molecule is also comprised of many secondary structures, which are called as random coil structure, alpha helical structure, and the beta sheet structure. So all of these structures, they can be present in a single protein. And depending upon their content and their folding patterns, different proteins, they perform different kind of a function. So my research is related to transforming this conformation from one to another and then developing a new biomaterial. That's what all of my research is all about, actually. So for that, we can see the secondary structure of the protein by many techniques, and one of the technique is circular dichromatism spectroscopy. And here you can see these are the different proteins like gelatin, collagen, albumin, lysozyme, and chymotrypsin. They give different kind of a secondary structural conformation in the CD spectra. This is because of the fact that that proteins are chiral molecules, and because of their different folding patterns and amino acid sequences, they absorb the circularly polarized light in a different way. Because of that, they show a characteristic and different kind of a secondary structure. However, all of these structures can be interconverted into each other by changing the physiological condition like temperature, pH, or the chemical agent. So my research is about interconversion of this conformation into each other and then finding the changes in the functional property of the protein. How does it changes? So this is my first work that I'll present over here is my PhD work, where we studied the conformational transition of cytochrome C. Cytochrome C is actually a important enzyme in the electron shuttling in the mitochondria of the respiratory chain. Okay. So yeah, so we use a bilayer membrane of an ionic liquid, which is having a structure similar to the structure of the cardiolipin present in the inner membrane of the mitochondria. So we wanted to biomimic these interactions, that how the cytochrome C, that upon interaction with the inner membrane of the mitochondria, changes its conformation. So we wanted to mimic them in vitro using an artificial membrane. And we observed was that the cytochrome C underwent all alpha to alpha plus beta conformational transition, actually, and which is also important indicator of the cell death or the programmed cell death in vivo. However, other than that, since we wanted to use it as a material, so we studied the change in the structure of the enzyme other than the conformation, like the size And you can see that at this particular concentration where the bilayer structure is observed, its size increases slightly actually. And because of that, there could be exposure of the active site of the enzyme to the substrate in a higher number, which led to the increase in the activity of the enzyme. So we see from this one is that just by changing the conformation of the enzyme, we can change its functional activity. This is one of the examples. This is another example where we use cytochrome C as a model enzyme as a model enzyme to study its thermal stability at high temperature. Yeah, because nowadays biocatalysis is a very important field actually, but enzymes they mostly work in the aqueous medium. And if we want to speed up the biocatalytic reaction, then we need to increase the temperature. But enzymes are benign to degradation beyond 50 degrees Celsius. For that, we needed a medium which can stabilize the enzyme beyond 50 degrees Celsius. For that purpose, we use cytochrome C as a model enzyme. And then we use an ionic liquid formulation comprised of a solvent ionic liquid and a solute ionic liquid, which form the bilayer structure inside the ionic liquid. And when we added cytochrome C into this one, this led to the higher thermal stability of the enzyme. And we studied the thermal stability using the CD spectroscopy over here up to 100 degrees Celsius, because CD has a limitation that it cannot operate beyond 100 degrees Celsius. But in situ, we heated the formulation up to 180 degrees Celsius for 5 minutes. And then we studied the activity of the enzyme, and enzyme activity was retained even at 180 degrees Celsius, which is a good indicator if you want to use biocatalysis at high temperature in a thermally stable media. And more than that, the activity was higher as compared to that in the buffer solution, which is indicating the fact that there is a conformational transition of the enzyme, which led to increase in its activity compared to the native one. Yeah, the third example that I want to show you, this is our recent work, which is from CICECO-Aveiro University when I was a postdoc over there. And it is related to the prompt or instantaneous fibrillation of egg white proteome using macromolecular crowding media. Actually, protein fibrils, they are basically known for their role in Alzheimer's diseases or the Parkinson diseases. However, they are also very good materials because of their functional nature and high surface area. And they have application in biotechnology and in other fields. But broad scale applications are restricted by the fact that it is very difficult to prepare protein fibrils in the bulk amount. And it requires very long time to prepare the protein fibrils. However, we developed a process by using egg white as a protein source, which contains a lot of protein, and that can act as a macromolecular crowded media. And when we added this ionic liquid into this egg white solution, there's a fast preparation of protein fibrils, and which was also evidenced by all alpha to cross beta structure, Scandi structure transformation, because the protein fibrils, they're always having a cross beta structure. So this is a structural evidence of that. And here you can see this is egg white, and upon addition of the ionic liquid, it transformed into a protein fibrils. Furthermore, we also studied its application actually, and we use these fibrils as an enzyme support for the biocatalysis, and we got good results. We observed higher activity of the enzyme upon when we use protein fibrils as an enzyme support. And moreover, we also grew some cells, like N2 cells, and using that, the protein fibrils also acted as a 3D matrix for the growth and culture of the cell as a cheap material, in fact. So we learned from here that by just changing the conformation of the protein from one into another, we can improve their activity or their functional activity. This is the work which I did as a postdoctoral fellow when I was in Japan, and it is related to photonics application of the protein, and how by changing the conformation of the protein we can achieve desired photonic application of a photonic property actually. So the work is related to triplet-triplet annihilation based photon upconversion in a protein. This is the general introduction of the TDOC, that it is comprised of a multi-chromophore system of a donor and acceptor molecules. And when the donor is excited at a low energy, it transfers its triplet energy to the acceptor molecule. And when two such excited triplets, they collide in a space-time, they form a high energy singlet state, which emits a delayed anti-Stokes fluorescence. So the photon upconversion is about exciting at low energy and then getting the high energy. So this process has application in the photovoltaics and also in the bioimaging, because in the photovoltaics, by using the photon upconversion, we can utilize the wasted sub-band gap photons and upconvert them into band gap responsive light of the solar cell to increase the efficiency of the solar cell. However, there is one problem with this process, that is that the triplet state of the molecule that can be quenched by the molecular oxygen present in the surrounding. So most of these processes, they have to be carried out in the deoxygenated environment. So to counter these issues, we use a protein, gelatin. So gelatin at low concentration exists in the form of a random coil structure, but at high concentration, it transforms into a gel or a film, and then it acquires a triple helix structure. So this triple helix structure is a good oxygen barrier. So we co-assembled gelatin with the chromophores, and we managed to prepare a hydrogel. And this hydrogel, upon excitation with a laser at low energy, emitted a high energy photons in the visible region in the presence of oxygen. So by doing this, we could develop a process where highly efficient photon upconversion could be achieved in the presence of oxygen. And the annihilator also showed a very long triple lifetime of 3.2 milliseconds. And this is the best upconversion efficiency, equivalent to the best upconversion efficiency obtained till date in the oxygenated environment in the aqueous medium. So by exploiting the conformational transition of the protein, here we observed a very good photonic property. So this is another work of ours, which was done in CICECO-Aveiro also. And here also, we use one protein's properties to stabilize another protein. For example, we used gelatin to stabilize R-phycoerythrin by using by developing a protein cohabitation concept. Basically, R-phycoerythrin, it is a part of photosynthetic antenna of the algae, where it acts as a photoreceptor, and then it transfers the solar energy to the reaction center to complete the photosynthesis. And it has an application in the photovoltaics and in the food, actually, as a food colorant. However, it also has a problem of the poor photochemical stability in vitro, which occurs because of the fact that when R-phycoerythrin is exposed to sunlight, it changes into triplet state, and triplet state further interacts with the molecule oxygen to develop a singlet state, which causes the photobleaching and the quenching of the fluorescence of R-phycoerythrin. So it is not stable in the sunlight for a longer period, or it is poorly photochemical stable in aqueous solution for a longer period. So we utilize gelatin for this one, and we again use the conformational transition concept over here, because gelatin in the form of film also exists in the form of a triple helix structure. And here you can see this is the model representing the fact that in the aqueous solution, R-phycoerythrin is poorly photostable. And since it is a protein, so it degrades within 2 to 3 days in the water. And if it is exposed to sun, then within 30 minutes or 25 minutes. However, when we prepared a film out of R-phycoerythrin using gelatin as a matrix by protein cohabitation, like this one. So we prepared films of pure R-phycoerythrin and the R-phycoerythrin extracted from the fresh alga. And we could get a very high transparent films, which showed, which are very, suitable for the optical application purposes or optical characterization. And then we did the time-dependent photochemical stability analysis using the absorption measurements and the quantum measurements for the 8 months. And here you can see the absorbance of the R-phycoerythrin in the gelatin matrix remain the same even after the 8 months. And also from the quantum yield measurements, the quantum yield remained the same after the 8 months. So by just changing the conformational transition of one protein, we can get a suitable material to stabilize that protein. So this was all about my presentation. So I want to thank the organizer for giving me the opportunity to present my research work. And finally, I want to thank the funding agencies, CSIR and DST India, GSPS Japan, ERC European Commission, FCT Portugal, and Marie Curie European Commission for funding my research. Thank you very much, everyone, for your kind attention.
Thank you, Pankaj.